Hi, boys and girls. Let's see. We're here with Dimiter and Rebekah from Bread of Life, and I will try to get that link to your channel in the description. And I don't know if everybody doesn't know you. Rebekah, who are you?
Well, I am a Christian evangelist. I have a channel called the Bread of Life channel. I recently wrote a book about evangelism in Muslim countries. And so you can find that at bridgesofloveandunderstanding.com. And so I have got involved talking also about evolution, because it's part of my story. And I think that's why we're kind of connecting here today.
Okay.
Yeah.
Dimiter, list all your credentials. Your credentials.
I'm a molecular biologist, and I'm working experimental science in the laboratories. And also I changed several fields in my career, doing experiment from variety of different topics, including cell cycle, DNA replication, DNA repair, cancer research, and now I'm stuck with the stem cell research. So basically, biologist and scientist. That's pretty much good enough, so to say.
Okay.
Yeah.
So I'm going to shut up. I'm going to let you two talk or figure out what you want to talk about. Is that fair?
Great.
And I will try to, I guess if I run into a question in chat, can you both see the screen?
Well, yes, I can.
Okay. Mm-hmm. So I'll put up I'll highlight the question so that you guys can spot it, so I don't have to try to interrupt you. And if you guys kind of keep an eye on, if there's any questions for you, maybe try to answer them, and we'll see how that works. Okay. But I'm going mute. You guys have at it.
Sounds good. Thanks, Mike. I like your bird, Dimiter.
Yeah, he's an African guy. He's small, but he's very eager and always kind of seeking for attention. And my problem with this bird here is that he's extremely social. It's painfully social. He likes people more than people likes people. So, and if you are not around him, he start to be unsettled, start to screaming. He even do not allow you to go out from the room and try to scream, where you are.
Wow.
So if the people around him, he's happy, especially if he have more people around, and then he's blossom, immediately start to pay attention to everyone, cheerful.
What's his name?
Tootsie.
Tootsie?
Tootsie, yeah. So cute. This is, anyway, that's stuck somehow. Somehow is that.
So, Dimiter, you sent me some papers on the origin of life. I read one of them. It's been now a few months since I read it. But what is it that you want me to understand about the origin of life?
Oh, okay. So I would like definitely to discuss that, and we'll come to that point.
Okay.
And I will discuss a lot about that. So, but let me first, I would like to ask you some questions because I'm
Sure.
also curious. Because um, from discussion, my actual encounter with the, let's say, evangelical creationists,
Mm-hmm.
I was always wondered what is all about. I mean, think about that. So, a book which is written very long time ago,
Mm-hmm.
uh, maybe 4,000 years ago or 3,000 years ago, have written something, and why that should be a problem for the modern science. I mean, the modern science established something as a
Mm-hmm.
fact, uh, mechanisms, everything,
Mm-hmm.
and why believing or not believing, let's say this way, in evolutionary biology should something to do with your faith in the book. I mean, it doesn't make any sense to me. So can you explain that?
Yes, I can. And so I would say, like, it's not for me, it's not a rejection of science. I believe that science is a good way to discover truth about the world. And I also believe the Bible. I believe the Bible's a true story. And so, when what I'm what I'm doing is trying to take in these two sources of truth and understand what they're telling me about the world. And so, um, I am not disputing, um, I'm not like creating this conflict between the Bible and science, but what I'm disputing is the some of the views that currently are being held within science. And I think that's a scientific thing to do, right? To like examine what is being presented to you as as a truth about the world, and to look at it critically, question it. And that's what I'm doing with evolution. I don't think that Darwinian evolution is a good explanation for why we are here, how we got here. I honestly do not think it is good science. Now, I know that the majority of scientists right now disagree with me and they have more expertise than I do. I'm not a scientist. So, I respect that they have more work in this area than I have, but there are scientists who uh, do not believe that Darwinian evolution explains how we got here. And so I take that minority view, and I think the reasons for that view, for taking that view, are sound. And I wish that people would think more critically about evolution. For me now, it's to the point where even if I wasn't a Christian, I wouldn't believe in evolution because I don't think it is, uh, like I said, a good explanation. I really don't. And so, you know, I lots of Christians have no problem with evolution and meshing it with the Bible, and that's fine if they make those two, and they don't want to like basically get involved with that, or even some of them are involved and they still believe evolution is the best explanation. So they kind of mesh that with the Bible. I don't think that evolution is um, the part of the clearest reading of the biblical narrative of creation. I think you can fit it in, but I think it's a little bit difficult to do that. So I think I have two good reasons for not believing in evolution. One, I don't think it's good science, and two, I don't think it really matches with the biblical narrative.
Mhm. So about the several scientists which does not agree with evolution, I will discuss as well, because from the position of PhD and working field scientist, which is directly involved in the actual real experiment, I can clarify that. So you'll be a little bit more enlightened for what is going on there. I want to ask you something else.
Okay.
Take Genesis, the first several chapters. Do you think this is a scientific journal?
Which journal?
The Bible. Do you think from
No,
the Bible
absolutely
Genesis
not.
is a scientific article or journal?
Oh, Genesis. I'm sorry. I thought you said
I'm talking.
geneticist.
Okay,
Yeah.
Genesis. I'm sorry.
Yeah, no, I don't think
Mispronunciation.
Genesis is science. No,
So
it is a
it's
story.
not a scientific story.
No, not a scientific story at all.
Why? Just explain to me why you're reading this Genesis story as a science.
I don't think I am. I'm
Yes,
not
you are.
reading it as science. I'm reading it as just as a story about our origins.
Well, the scientific papers usually have experiments or even hypotheses. There is a graphics, there is a data, those kinds of things.
Exactly.
And I don't think Genesis it's actually possess any of this. So what is all about Genesis that it's attractive to you?
Well, we agree on that point, Dimiter. I'm not reading Genesis as science. As I said, I believe that, okay, a scientific paper, that's a good way of discovering truth about the world. I also happen to believe that the story in Genesis is true, but I'm not calling that science.
Well, okay, so you
But
believe
what's
it's
attractive?
true.
Mhm.
You believe it's true based on what?
Well, based on quite a few things. First, I think it's the best explanation for how we got here. I don't think that science explains where matter came from, why we have a universe. I mean, you can say the Big Bang, but if you say the Big Bang, that still doesn't explain why the Big Bang happened, where in the, how does this event occur, right? And so, there's like I believe that the story in Genesis is a good explanation for where we came from. We have a creator. God created. God created us with intention.
So who told you that we scientists should know everything? We also don't know. We don't know lots of things.
Yeah.
Rebekah, oh poor Rebekah, we're just human beings. We are not gods. We don't know everything. That's why the science exists. I mean,
Yes.
the reason for existing science is because we are trying to look and trying to figure it out how the things are working and what actually the explanation for things are. It's nothing to do with we must know everything. I mean,
Yeah,
we
I
don't
agree.
know a lot. We don't know a lot. I am curious about so many things that I don't know how it happened, including origin of life. We don't know how actually happened. But that's not the idea. So the idea is that the science works in different way. The science works with the most probable explainable way. If something is most probable and if you have real explanation for it, that's more likely to happen. So if you say, oh my goodness, are you absolutely sure that exactly that happened? Well, that's not the science. We know a lots of things that exactly how it happened. We know a lot, but that doesn't mean we know every single bit of what actually happened. And also, if you look the history of science, we're pushing the boundaries. I mean, look the science in, for example, 12th century. 12th century science is all about surrounding us. The world was pretty much from Spain to, for example, there's a cracking sound coming from someone.
Yeah, I
Yeah,
think it's coming from you, Dimiter. I'm
think
not
it's your
doing
bird.
anything.
I think it's the bird.
I think it's his
No,
mic.
it's not.
I think it's
Yeah,
the
the mic.
microphone.
Is it now okay?
No, it's good. It's good now. I think you just have a connection problem or something. But go ahead. Sorry.
Let's try just for a second. I will unplug my. So let's unplug my.
I think if you move around, I'm not. Can't hear you now.
Can't
Oh, you're
hear you.
muted. You're muted, Dimiter. Unmute. Oh, I muted you again. Sorry. Try now.
Okay, what about now?
It's pretty good. It's a little tiny bit of an echo, but it's much better. So no, actually it's really good.
Yeah, it's
It
better
is
than the
better.
crackling.
Just
Okay.
give me 1 second to adjust my own sound, and that will take just a few seconds.
Yeah, I can turn you up a little bit too, I think. Let me see if I
Well,
got you.
I want to listen. I want to listen better. So,
Oh, you're adjusting it. I think you're good.
just no, I want to listen better. So, let me see. Okay, so I think if you're listening to me, I also can listen well. Okay.
Okay.
Are we good now?
Yes.
We're good.
Okay, small technical problems. So, okay, so where was I? I was trying to explain that. Okay, back to the. So if you actually see the science in 12th century, for example, the entire world was, let me say, from Spain to India, something like that. Everything else does not exist. And if you go further, 18th century, now we have New World and so on and other stuff. If you look further and further, if you go even the beginning of 19th century, we even didn't know what the galaxies are. We even didn't know where the galaxies are. Is this the entire world? So
True.
the working of the scientists, like Hubble, for example, he actually discovered that there is other galaxies. And so on until now, 21st century, we know from Hubble telescope that if you go further, there's still you can see galaxies. We don't know where the boundaries. We know the boundaries somewhere. Is there any boundary? We don't know. There are lots of questions. We have no idea.
Mm-hmm.
But let's go back to the Bible for a second.
Okay.
There is a good science in Bible, and I can tell you, it's called textual
criticism.
manuscript. Yeah. So, and based on this, we also can see the evolution of Bible. So it is like a phylogeny of the Bible. So the different texts and manuscripts, if you find the best manuscripts, you can see how the Bible is actually under development. You can see several layers in the Old Testament, for example, at least 5 to or maybe more layers, written in different times, written from different people. Now you can argue how they're inspired from God or not, but I can see here a little bit more human work. So the humans actually write down the Bible.
Absolutely.
Let's go back to the New Testament.
Mm-hmm.
So have you ever heard about Q gospel?
Yes.
So you know what the Q gospel is, right?
Yeah,
So
it's the
the Q gospel, because I like history, I was going in the history of Christianity. Of course, I'm not specialist and I'm not pretending to be a specialist, but as a scientist, I got a relatively good view about what happened. For example, Q gospel, it's a hypothetical gospel, which is about a written saying of Jesus before to be written down from Matthew and Luke down to their own story. And also, so Matthew and Luke get two actually common sources. One is the Mark, the very first the very first gospel, and Matthew and Luke also get in possession Q.
Mm-hmm.
Now they merge together in their own way, and you see actually how those two gospels come to be. And you can read actually recovered Q gospel directly. You can download it and you can read. This is the saying of Jesus. So we can assume that between the Jesus actual time and until the moment it's written everything, there was a kind of circulating Q gospel. Of course, they have their own sources. Matthew has its own sources, Luke has its own sources, and so on. We even can see exactly, not exactly, I'm sorry, approximately when the Gospels was written. So the first Gospel, Mark, was written around 60, year 60 probably. Matthew and Luke was written about 80s of the first century, uh, and the last one, which is John, it's written around here 100, 120. So, not to mention, there is also some gospels that didn't make it to the Bible, like a Gospel of Peter. So, Gospel of Peter didn't make it to the Bible and was written approximately at the same time, about 80s. Why the Gospel of Peter didn't make it? Because it didn't fit. And why didn't fit? Because Gospel of Peter says that Jesus didn't actually suffer on the cross, and that didn't fit with the major concept for suffering and redeeming, you know that stuff. So, bottom line, you see how compare the text shows emerging of very valuable texts in the past and the history of the actual emerging uh, of the Gospels. The very first text is which one in the New Testament?
Probably one of the letters of Paul.
Exactly, the Paul letters. So this is the very first text that we encountered is written in the New Testament. And Paul is not even disciple of Jesus. He even haven't seen it at all. Now, from the perspective of modern theology and so on, he was witness enlightenment and Jesus appeared in front of him. Okay, I'm not discussing that right now. But the fact is that he is not even disciple to Jesus. However, he met brother of Jesus for 14 days, according to the letters, in Jerusalem. So what they discussed during that 14 days, it's very interesting, but I don't think we'll know ever what they discuss. It's very interesting what they discuss. 14 days, it's a good time to spend with the brother of Jesus, right? James.
Mm-hmm.
Okay, the second thing. Tomorrow, probably you'll go in your Sunday school, right?
Mm-hmm.
Humor me. Count citation in the pastor. Every time they go back to the Bible and they put citation there, right? How many times the pastor cited Paul instead of everything else? I did that because I've been listening pastors probably around 8 years.
Mm.
I'm listening pastor mostly to the radio and also going in the church to listen that. And if you try to count, of course, I'm not saying that counting is something spiritual. No, that's not the idea here. If you try to count, you'll see that the pastors, evangelical pastors, they're using Paul in probably between 80 to 90% of all of the citations. This is the master of evangelical speech, nothing else.
Okay.
Now, try to recover the real historical Jesus events just based on Paul's letters. If you don't have anything else and you have Paul's letters, figure it out. What actually Jesus did? And you will see almost no encounter from the life of Jesus based only on Paul's letter. A few things, of course, will emerge. And this is the Last Supper, of course, and of course that the crucifixion. Pretty much nothing else. So now going back to the analysis, I'm not trying to do anything with your faith here. Everything I'm trying to say is think about the Bible in more humanized way. Is a real work. It's extremely important scriptures because the history of Europe and the entire world actually have, the impact of the Bible have a huge impact over the history. And that's why it's very important. But my point is just try to be open-minded.
Mm.
And also I'm trying to say something else, that spirituality not always mean alphabetically, literally understanding of the things in the Bible. And I think you will agree with me that there are lots of things, lots of texts in the Bible that could be accepted simply, not literally, but symbolically, if you wish this way. Forget about is it real or not. I'm talking only for real spirituality. That's why lots of scientists as well, in fact, are not atheists. Lots of scientists actually believe in God. But they also very well know that whatever is written in the Bible could be accepted, lots of things could be accepted simply symbolically. You don't need to accept those things because literally, you don't need to accept those things literally. It's very simple. I mean, someone wrote down 3,000 years ago Genesis, merging two different stories and make the Genesis. He copied, or she, I don't know, probably it's he, copy from somewhere. And we know very well that Sumerian and so on stories was influenced this. And that's very interesting. You see more human Bible. Yes, inspiration is your thing. You can think about the inspiration, that's your work. That's fine. But I'm saying you don't need to contradict science and whatever science discovered with the Bible. I can tell you one funny story also. A modern analysis, if you go back to the Hebrew Bible and the original wording of the Hebrew Bible, you know, according to Genesis and so on, the Eve was created from Adam's rib, right?
Or from his side.
From inside.
Mm-hmm. From his side.
So there was a modern, so the rib was very circulating all the time. But the modern analysis, which I read in the journal from Biblical Archaeology journal, there is a good analysis from biblical scholars, which says very well that if you take this word particularly and try to analyze the situation, you will see that in fact, Eve was created according to this scripture from the penile bone of Adam. This is the penis bone of Adam. You see what I mean?
Okay, I haven't heard that one
lots
before.
Lots of animals, and this is known back in time. Lots of animals actually do have penile bone, but the humans don't, and probably primates also don't. So this is an ancient explanation why humans don't have this particular bone. See, it's a funny story, but how is twisted the analysis, what they mean in the past and what is emerged after that.
Yeah.
So that's all about what I wanted to say about the Bible, and just provoke you and try to encourage you. Go out and don't think that everything in the Bible should be accepted literally.
Well, Dimiter, you're making a lot of assumptions about me. You know, I think you don't know that much about what I think about the Bible.
No, I don't. But the message is not just to you, but everyone that is listening as well. And I think you probably will agree with me.
You know, and I'm actually, what I'm impressed by is that you have read the Bible. And
I didn't read the Bible. I didn't read the entire Bible. Of course, I read lots of. Okay, the New Testament. Yes, I read the New Testament, but not the entire Bible. So, and I never claim myself a specialist to this. I'm not a scholar. Mm-hmm. Not a scholar. Just I'm interested in history of the stuff.
Well, see, what's interesting, and I'm very happy that we're having this discussion, and I'm happy to hear that you have, you know, read at least the New Testament, and that you've done work beyond that. You've kind of looked into what are the sources of these documents. And I don't disagree with a lot of what you were saying. I might, you know, dispute some of the dates you were mentioning, but scholars dispute those dates, so that's no surprise. But, you know, and even the existence of Q is debatable, but, you know, either way, I'm fine with Q or not Q. It's fine. But I'm impressed that you've done a little bit of work on that. But what's interesting is I thought we were going to have a discussion about your specialty, which is science.
Now we are starting.
Okay. Well, we might run out of time because I have to go like a little bit over and out. You know, I'm having a stream on my channel in a little while. So, and I hope whoever's watching here can join that because I'm interviewing a Trinitarian Muslim, and that I think that'll be an interesting perspective for people. But really, I agree with you that science does not have to be in conflict with the Bible. When I'm discussing the origin of life and evolution, I'm not doing it with the Bible in hand saying, no, the Bible is true. When I'm talking about evolution, and that's why if you look on the Examining Evolution channel I started, there is nothing about my faith on that channel. I even though I don't have a lot of time to work on it right now, so I've abandoned it. But if I pick it back up, my vision for that channel is science and only science. This has nothing to do with God. It has nothing to do with faith. We are looking at whether or not evolution is true by scientific observation and experiment. And
Okay.
so, you know, I agree with you that I don't need to, you know, have the Bible be in conflict with science. I'm looking at the science and I'm saying I don't think evolution is true.
Okay, so let me start with the scientists now and the evolution. So, and based on what I saw a few times in your channel and with discussion before, you are citing and you referred a few scientists from Discovery Institute. And what I'm trying to say, so now we're in my area, because this is the area of my expertise. What I think is happening here, and you have to pretty clear understand that. The scientists which reject the evolution not always present to you or to people like you the correct model how the evolution works. And that's a problem, because let me give you a very simple example to clarify that. If I say to you that there is a way to go to the moon, for example, you can say, no, you cannot go to the moon because you cannot jump that high. I cannot jump to the moon. And here's the problem, because yes, I agree with you that I cannot jump to the moon. Definitely, I'm not a Superman. But I'm not presenting this way of going to the moon. I'm presenting totally different way of going to the moon. Building a rocket, building a spacesuit, building blah, blah, you know, technical stuff there. So here, just to illustrate the situation, if you insist that the way to go to the moon is always jumping to the moon, obviously we'll disagree every time. And if you completely reject going to the moon with the rocket, obviously there is no way to go to the moon. So let's start with X. I'll give you 3 examples here. So you like X, right? Do you?
Do I like X?
Douglas X, yeah.
Oh, X. Oh,
Discovery.
Sorry.
So
your accent
let's
threw me
go
off.
directly to the science.
Yeah.
So let's go directly to the science and illustrate the situation, because you are misinformed. And once you are misinformed, of course, you will not agree. So Douglas X, I listened to this guy probably 3 or 4 years ago, don't remember. But anyway, so I remember at least his argument. So he was discussing in one of his videos, and probably he have more videos anyway. So he was in a real science before, maybe in Harvard or something like that.
Mm-hmm.
And he presented 3 stories: good, bad, and ugly. So the good. So his experiment was that during that time in Harvard, they was looking for the resistance of some antibiotics to bacteria. So, and the resistance is due to the enzyme which actually chew out and destroy the actual antibiotic. And of course, if the bacteria is capable to destroy the antibiotic, of course, will be resistant to this antibiotic, correct?
Mm-hmm.
So his experiment is quite valuable. So what they did mutations in the gene of this particular enzyme, which starting with very small mutation, like a point mutation, which you delete just 1 or 2 nucleotides or change some nucleotides. And he see that there's not much effect on the resistance, meaning the bacteria still possess resistance. And to him, he said, okay, look, the evolution can adapt a little bit. You know, the dogs can be a little bit this way, the another way. So the bacteria can adapt a little bit, and that was good. And he accepted that, okay, the evolution have some small adaptive features. So correct me if you understand differently the situation. So I'm trying to follow
No,
his thought.
that's fine. Mm-hmm.
So, and he tried to delete a little bit more from the gene, the same enzyme that produces the same enzyme. So now we have a problem, but sometimes actually the bacteria could recover, but in a more difficult way. So, and he said that, okay, now there is no de novo information, cannot come de novo information, and the bacteria have difficulties. Let's delete the entire gene. And he deleted huge portions of the gene, which we can do every day, those kinds of things. You can delete the gene, you can add gene. We do that all the time in our experimental science. And now the bacteria cannot have resistance at all. His conclusion was that there is no way in the nature to make new information. Therefore, look, this is experiment. There is no appearance of the gene that can overcome the resistance from nowhere to come this and to generate again resistance. Therefore, the nature is not capable to make new information.
Okay.
Am I correct for the vision of Douglas X?
Well, I mean, you're simplifying it, but
Of course, I'm
yes.
simplifying and intentionally simplified, but that's pretty much the picture.
Yes.
So, and that's ugly. No new information, cannot appear new information. That's why it happened.
Yes.
Now, first of all, I want to say that the deletion mutants are something which we are doing all the time. So this is the way in science how we actually try to study the function of some genes and particular proteins by making a deletion mutants. And this is a hugely, hugely overwhelming papers, thousands, even not 100,000 of papers in deletion mutants, because this is the way how we study the proteins. Not to mention that we have a mice, which we know every single gene, is it vital or not? If you delete particular gene, you see the mice, is it going to be born or not? Is it going to survive or not? We also know from the entire set about 30,000 genes, which gene is actually essential for the cell. Always based on the same approach, deleting a gene or deleting a portions of the gene. Now, he published the papers from the perspective of looking into the resistance, of course. But what actually he don't tell you that the evolution does not work in this way. This is not the model we are working.
Okay, what is the model you're working with?
He misrepresents the situation.
Okay, what is the actual situation? What is?
Okay,
what is?
that's exactly what I'm trying to say. Of course, the bacteria or any other organism cannot recover the gene when the gene is deleted. That will never happen because it's not possible. What and how the evolution actually works, if you have a mutation, you can go in different selection now. Something that he does not present it. In fact, the evolution does not work in a way that you have mutation and now the organism is obliged to recover this mutation. That's never happened this way. You have mutations, but also you have different selection, environmental selection. Sometimes it happened that this organism, let me say the bacteria with the deleted gene, might be seeded in different environment, in different area where this deletion might have an advantage, not disadvantage. And the reason for this is because in particular different environment, different selection, now we have different conditions where another set of mutations might come up more advantages. This is the actual model of evolution. It's not about making from nothing new information. Now the information can come, new information can come, but not based on the way they present it to you. See?
Okay, then in what way can it come?
Okay, I just want to point out the misinformation that has been pointed to you. Now, understanding the actual model, how the scientist works in the informational or evolutionary biology, now you see that you were misinformed. This is not the way how it was working. Simply,
now, Dimiter, I wrote down some quotes from you, okay? And this, and you said once it cannot recover. Once a gene is deleted, it cannot recover, right? It can't come back. But you're saying, and you said it can never happen. Okay.
No.
So that is what you said. Now, if you want to change your what you said now, that's fine. But what you're expecting me to believe is that thing that you said can never happen, well, that actually had to happen many times. You had to not just recover a gene, but you had to get a gene that never existed to suddenly appear, right?
Okay.
And you said you have different selection, but where did those things come from that we have a selection possible? And this is the main thing that the problem with evolution is that you always do need something to select from for natural selection to work, but you are never giving me the mechanism by which, as Douglas Axe says, the de novo information can arise. And you've got to get that for the origin of the very first life, which is I don't know. We have not found that. We haven't even found a path for that to happen yet. And then even once life arises, then you've got to get all this information from somewhere before nature can select, right? So you're saying this is not about making new information from nothing, but it absolutely is. That's the whole point, is that where did all this information come from? Where did the information for making hands, eyes, all the organs and systems that we have in our body, where did that information come from? And you're telling me somehow it arose by chance, by just, a blind natural process. And I'm saying that is absolutely ridiculous. When do we ever observe that happening in all of our scientific observations, in all of our scientific experiments? Where do we see that kind of information arising? If we tried to make a human heart in the laboratory from scratch, from nothing, can you imagine how much we can't even do that with all of our intelligence working together? But you want me to believe that arose spontaneously by time and chance, and that makes no sense. Please explain to me how this type of thing could occur when we can't even working together with all our intelligence create something like that. And we need that to happen millions of times for all the things that we see in life and the way that plants, humans, animals.
I'm sorry that I disrupt. I got the point, but we don't have much time. So I got the point, and I see your confusion. So first of all, I want you to agree with me and to understand that Douglas X misinformed you about how the evolution works. He didn't present the correct model for evolution, and he didn't present that, in fact, you must put different selections, different environment in order to undergoing evolution. And I'll explain about the information just a little bit later, but let me be sure that you understand this point. Because we scientists does not work in this evolutionary model. We do not expect something to arise because it's not possible.
Okay.
Now, introducing a wrong model in your head, it makes a confusion.
What is the model?
So, okay, I told you, you have different selections. So I'll tell you about the information, how it comes to be the information. Now, I just wonder where to start, but let's start from somewhere, and that is the genome. We know all the mechanisms of arising of new information. How that happened and what does it mean? New information is not new in a way that it's completely rewritten from scratch. New information is a copy of prior information because you have amplification processes.
Where did the original information come from?
We'll come to that point just step by step, because I cannot describe the entire evolutionary biology in 5 minutes. That's not possible.
But what is the mechanism? Where does that...
Exactly, that's what I'm trying to tell you.
Okay.
So this is amplification. You have recombination. You have mutation. This is 3 major events which we observe every day in our laboratory work. All of this is observed every day. This is not something that I need to prove to you right now. Let me assume, just agree with me, that there's thousands and thousands of papers on repair mechanisms.
I agree.
And now, if you have a gene, one gene, which is amplified, and let's make 2 identical genes. Now, this amplification occurred because of process of replication and, all of that repair mechanism, which right now I'm not going in details. If you amplify and you have duplicate one gene, now the cell contains 2 genes of the same genes. However, most of the time, the second copy might be put somewhere in the genome, the other place in the genome. And now you have a situation where you have another copy of this gene somewhere in the genome, which is stay there. However, the mutations is undercoming because the second copy of the gene is not so essential, meaning the first copies do the entire work. The second copy might be just additional, but if it deletes something, if it mutates something in this particular second gene, nothing will happen in terms of survival of the organism. It's simply amplified. Sometimes you can have phenotype, you may not have phenotype. Now, think about that. You have a further recombination events, and we're observing that appearing of new recombinations among the genes. Now, that's why if you look every single gene and vice versa, every single protein, you will see that the protein is not built up in unique sequence. Every single protein have homologs somewhere. That's why we call family of the proteins, because every single protein is made up from domains. They can rearrange. In fact, there is approximately 200 domains of different proteins from the different proteins, but this is a smaller part, a functional part of the protein. You have domain, for example, for ATPase activity. You have domain that is responsible for DNA interaction. You have domain which is responsible for binding with another domain. You have domain that is responsible for phosphorylation of another. This is a small domains. If you recombine those domains, you have new protein. Although new protein doesn't mean rewritten new from the scratch. This means recombined protein. In fact, the entire biology is recombination from prior stuff. So you have a building blocks. So if you go from organ, for example, organ, and compare with another organ, let me say, whatever, kidney with lung, for example. In fact, if you think about this, is two different organs, they're doing totally two different things, correct? And that's true. However, if you go inside, and you'll see that every organ is built up from vasculature, you have neural tissue, you have endothelial tissue, and all of the organs have a specific cells which is doing the job, but you have additional different tissue which is the same. In general, this is the same tissue in every single organ. Every organ is composition of different tissues, but those tissues are the same everywhere.
Okay,
Like a
Dimiter.
glandule, for example, if you take a glandule, there is 100 different types of glandules, but they have the same composition of glandule. You have a lumen, you have secretory cells, you have a capillary system, and blah, 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 lots of things.
Mike, I can go for like 10 more minutes, but you're unmuted and you're really loud.
I am?
Your background is like really loud. Dimiter,
Really?
can I respond to what you're saying? Okay. You mentioned amplification. Great. I have no problem with that. Recombination, no problem with that. Mutation, no problem with that. Right? But what I want to point out is that all of those things, amplification, what does it need? It needs some information to already exist. Recombination, what does it need? It needs information that already exists. Mutation, the information must already exist. So in all of these processes that you're describing, you're starting with information. You said, that the entirety of biology is the recombination of different stuff. But what I'm saying is, where did the stuff come from? And that is the main problem. I agree that the stuff mutates, right? But the question is, does it produce these new structures? Does it produce? And we do not see that in scientific experiments. We don't see that in our observations of nature. We just don't see that. And so all of these things that you're describing, I have no argument that, all these processes happen: amplification, recombination, mutation. No argument against that. So I accept all those papers that you're talking about. But where did that information come from? And that is the main problem here. So what I'm asking is, where did the information come from?
Okay, so this is about evolution now. If you compare organisms everywhere, you will see, and you can build up so-called phylogeny network based on comparison between different organisms. And you can see there is simple organisms and they are more complicated organisms. And also you can see that the part of the simple organisms can be found in more complicated organisms. The major rule, which fit exactly with the evolutionary theory of the evolution, is that if something is extremely important for surviving of species, it will stay more conservative in the time, meaning will be much like the same every time. And that's exactly what we see. The conservative regions are more important. Everything that is important is more conservative. If you build up projection back from the phylogeny network, you will see that you have a simple and simple and simple organisms and more simples. For example, archaea is much more simple than the common eukaryotic, higher eukaryotic cells. Or bacteria is more simple, and so on. If you build up such a network, such a tree of phylogeny, which is absolutely real tree, you can compare how it's possible you to have the same gene with bacteria, for example. Is it possible? Yes, it is. And this is mostly ribosomal part. And mostly those kinds of things which is essential, absolutely essential for life. That's why we see the picture how the evolution goes from simple to more complicated. It fits perfectly well. Not to mention that we have completely good picture about those mechanisms by building up and see how the family, you have also phylogeny of the family of the different proteins. That's how the things are going. You cannot judge something because I'm saying something about higher eukaryote. For example, what is the situation in the human? Human contains probably about 50 different polymerases for DNA, and they have different functions. But the bacteria contains only, for example, 3 or 5 polymerases. So you see the things. Although the polymerases can appear several times, that's a different issue. But the principle is the same. You can see how the phylogeny is going. Meaning, you have much simple organism at the beginning somewhere, which actually go and start to build up and make it more complicated organism. And that's how the information from more simple information goes to more complicated information, because you have amplification, selection, and selection. Mutations, selection, amplification. You can see, for example, let me say as an example, you can take keratin gene. Well, there's at least 50 or 80 different keratin genes, and all of them are eroded in the same section. So how that happened? So many different I mean, they are the same genes mutated slightly. It simply was amplified. But now they pick up different function. Some genes are more suitable for this way, another genes are more suitable for this way because of the selection. And then it's coming the question, okay, if you have a very, very, very simple organism, which you said you have to have information for that, how that come from, right? And we know that everything that we have is alive at the moment have a root somewhere in the bottom, which is called last universal common ancestor, Luca. Why we know that? Because of the phylogeny. Because Luca contains the same absolutely necessary gene for ribosomal genes, for metabolitic genes, for some of the replication genes, some of the cellular, let me say, membrane containing genes. It's a common. You can compare them, and sometimes they are very conservative genes. Therefore, we have pretty good idea about the genome of the Luca. The genome is approximately 300 genes of this particular Luca. It's very simple. And based on the selection and mutation, as I described, you have all of this multi-universe of so many different species. In fact, there is not much different in general. So, meaning you have always some building blocks. You can come up everything. The entire biology comes to 20 amino acids and about 5 different nucleotides and several lipids. Everything that you know, it's about that. So having this and this multiple diversity of organisms makes perfect sense. If you buy a Lego game, you know the Lego game, probably from eBay, you can buy a huge bag of Lego parts. You can build castle, you can build a car, you can build a tree, but in fact, all of this that looks so different are built up from the same building blocks. And now we're coming to another guy from Discovery Institute, which is the mayor.
Stephen Meyer.
Yeah.
Yeah, Dimiter, is it okay if we save Stephen Meyer for another day? Because I'm going to have to go, but I'll just let you have that last word there. So thank you so much.
So are we willing to do a part 2?
Sure.
Okay, we'll do it. Let's
go next week. If you want, next week at the same time, we can meet and discussing directly for origin of life and emergence of information. Because now you saw how actually the things are working. Now your question is how the information came to be at the beginning. And this is the Stephen Meyer's misunderstanding, which also present a wrong model, and also James Tour misunderstanding and provided a wrong model. And by working and providing wrong models makes people like you completely confused about how we scientists talk about evolution.
Okay. Let's
I don't know if I can do next Saturday because I have another stream next Saturday, but let's work something out by email.
Are you dodging Rebecca?
No, no, not at all.
Okay.
But
we'll
actually
figure it out.
Okay, we'll figure it out.
Okay, text.
Okay, thank you guys.
Thank you.
Thank you so much.
Goodbye, cruel world.
Bye.